Kia ora koutou katoa, it's Mrs. Goodson here, and this is a video about planning a science investigation and how to set up the variables and write a hypothesis for your science investigation. So, what is a science investigation? So, it is a way of collecting observations and evidence to test a theory or a hypothesis in a very organized and careful way so that it is a fair test. The aim of an investigation is the question we want to answer. And to make sure the investigation is valid, to make sure that it is a fair test, we only change one thing or one variable at a time. The one thing we change is known as the independent variable, and we keep everything else the same. So, this is a way of remembering the names of the variables. So, cows moo softly. Change one variable, measure something, and keep all the other conditions the same. So C for change, M for measure, and S for keep the other conditions the same. So there are three variables that you need to remember the name of. So the independent variable is the thing that is changed. We only change one thing so that it's a fair test. The dependent thing is the thing that is measured, and the controlled variables are all the things that are controlled or kept the same. Here's just a graphic representation of that. So in this little experiment here, the plant is growing. The thing being changed is the drink or the fluid being added to the plant. The height of the plant is the thing being measured. So that is known as the dependent variable, the thing being measured. And everything else is constant, is the same, or remains unchanging. So the type of plant, the pot size, the amount of liquid, the soil type, all those other things that might influence the results are kept the same. So what is a hypothesis? So the aim is what you're trying to investigate and should refer to the independent variable. The hypothesis is a testable theory or a testable hypothesis linked to the underlying science idea. You must be able to say whether you accept or reject your hypothesis at the end of the experiment. You cannot say that you're kind of right. So your hypothesis has to be carefully worded so that you can say yes or no, you accept it or reject it. So you could start with, I predict that changing the, whatever it is, will have the effect of increasing, decreasing, or keeping the same on the dependent variable, and then because is the link to the science idea. So we're going to do a practice investigation for our biology internal, and then we'll do other science investigations. So we've got our aim. In this example, does the amount of sunlight affect the height of radish plants? So in our aim, in this example, the amount of sunlight is the thing that is being changed. So that's the independent variable. And the height of the radish plants, in this case, is the dependent variable. So you could write a hypothesis like this. I predict that as the amount of sunlight increases, the height of the radish plant increases. Okay? So that would be an example hypothesis. The independent variable, the amount of sunlight, is going to be in minutes of sunlight per day. And we're going to test out 60 minutes, 120 minutes, 180 minutes, and 240 minutes. Okay, so that's called our range. So that's how we're going to change it, and we're going to have 4 different data points in our range. The dependent variable is the thing measured. In this case, it's the change in height of the plant, and we're going to measure that in centimeters. See how we put the units in? And then we have the same type of plant, the same amount of water, the same soil, the same pot size, etc., etc. Okay, so that's just an example how you might set up a science investigation. Now, what about reliability? Reliability means that another person could carry out the same method and get the same results on any day. Okay, so to ensure our investigation is reliable and gives trustworthy results, we're going to carry it out 3 times. And we're going to take the average of those 3 trials. Now, it's important that our trials are as reliable as possible. So if we get some data that really doesn't fit, we are going to say that that is an outlier and leave it out. And we either repeat the trial or we just leave it out of our average. Okay, so we're going to do at least 3 trials of each independent variable. Now here is a good video about fair testing investigation, another example for you to look at. And so this video has been about identifying the independent variable, the one thing to change, the dependent variable, the thing to measure, the control variables, the things to keep the same that might influence the result, so that our test is, we control these variables so that our investigation is a fair test and it is valid. How we're going to make it reliable by repeating our trials, and how to write a hypothesis.